Thanks for coming to my talk. This is so nice. I'm really excited. I'm going to be sharing a study I did. It was my last chapter of my dissertation, where I looked at plasticity of larval dispersal-related traits in the orange anemone fish. So I know this is a symposium for the interplay of dispersal, plasticity, and adaptation, but I just wanted to specify when I say the word dispersal, I'm talking specifically about the movement of individuals away from their natal location, or in other words, just babies moving away from their parents. So a really useful way for us to measure dispersal is as a dispersal kernel, which is a relationship between the probability of successful dispersal on the y-axis and the relationship with the distance traveled on the x-axis. And this is a very classic example of a dispersal kernel where we see successful dispersal declining pretty exponentially with distance. And I'd hope this would make sense, right? If your parents were able to live somewhere and well enough to reproduce, it would make sense that you would want to stay close to your parents because it's probably a good spot. Versus the further you move from your parents, the more likely you are to maybe land in a bad habitat or completely inappropriate habitat. Now, dispersal plasticity is the ability of an organism to change its dispersal phenotype in response to an environmental cue. So here we have another wind dispersing plant, but it produces two different morphs of seeds. So when the parent plant's in a really good habitat, it will make these seeds that just drop and they'll germinate really close to the parent plant. Versus if this plant finds itself in a really poor quality environment, it will switch and it will make seeds that have this ability to disperse with the wind, right? In theory, to be able to travel further away from that bad environment. All right, so a dispersal kernel for a species with dispersal plasticity might look something like this, where you still have that one dispersal syndrome where most of the individuals are staying close to home, but then you might also see this other dispersal syndrome shown in orange, where individuals might have a higher probability of dispersing those greater distances. All right, so here a dispersal phenotype can be seen as an adaptive phenotype, and in the case I described specifically, as an adaptive parental effect. All right, now dispersal plasticity as an adaptive parental effect has found widespread, and especially in terrestrial systems, in many plants and invertebrates, and even vertebrates such as these birds and lizards. However, it's only been hypothesized in marine fishes, which prompted my overall research objective. There should be a picture of a fish there, but is there dispersal plasticity in marine fishes? Okay, so first, to explore the possibility of dispersal plasticity in any system, we initially need to think about whether the conditions for it to evolve are met. So very, very simply, if you will, we can think about it as these three criteria, right, for dispersal plasticity to evolve as an adaptive parental effect. So first, parents need to experience variation in the environment. Second, those parents have to be able to reliably assess or predict the environment of their offspring. And third, the parents have to then have the ability to alter their offspring's dispersal phenotype. So to investigate the possibility of adaptive dispersal plasticity in a marine fish, I used Amphiporon percula, the orange anemone fish, or clownfish, or probably widely known as Nemo, and it's become a model system in marine fish dispersal ecology, as it's one of the handful of marine fish species that have had its dispersal kernel empirically measured using parentage analysis. And we see this classic dispersal kernel, right? Most individuals are staying close to home. And then you have this exponentially declining probability of successful dispersal as you move away from parent populations. But we see this wide variation. Like some individuals are dispersing further, like up to a magnitude further. And currently, we do not know the causes of this variation. So other than thinking maybe dispersal plasticity could help explain this variation because it's found in so many other systems, we have other reasons to think it might be in the clownfish. So these fish are demersal spawners. The males provide parental care, they hatch, and then they disperse as teeny tiny little larvae. And then if they're lucky and they don't get eaten, they'll settle in another anemone away from their parents' anemone. And then that's it. Then they do not move after that. As adults, these fish do not move. So you would think having an informed dispersal strategy would be really important in this system. So back to our conditions that we need to first establish when looking for this. And this first condition, there's a lot of prior evidence that their parents are experiencing variation in environmental conditions, such that this high variable habitat qualities confer an effect on their fitness. So next, we had to address whether parents can reliably assess or predict the habitat quality of their offspring, right, in order to correctly and appropriately respond in such a way that it's adaptive. I addressed this in a previous study that I'd be happy to chat about more, but essentially I found positive spatial autocorrelation in multiple habitat quality indicators. So at our study site, it roughly translated to parents are able to predict the habitat quality of their entire individual patch reef from their distinct location in their anemone. All right, this brings us to our third criteria, which is the experiment I want to share with you today, where I tested whether parents could do this. Can they alter their offspring's dispersal phenotype? So I used a laboratory population of anemone fish, and I measured dispersal-related traits of the larval fish just after they hatched. I measured swimming speed and their total length. Specifically, I asked, is there a plastic response in these dispersal-related traits in response to their parental environment? So I predicted that offspring that come from a really good quality environment, they would exhibit short dispersal syndromes, whereas offspring from parents in really poor quality environments, they would exhibit a long dispersal syndrome, right? In theory, to leave that bad environment and hopefully find someplace better. In terms of the dispersal-related traits, I'm expecting a plastic change between shorter, slower larvae and longer, faster larvae. So to manipulate the parental environment, I manipulated the parents' food rations. So I had this really high food ration or this very low food ration. We had about 34 pairs of breeding clownfish that were consistently breeding in the lab. So I put half of them, or 17, first on the high ration, the other half on the low ration, let 3 months go by, and then I collected 15 larvae from each breeding pair. And then I switched the treatments, right? So if they're on the high, then they're on the low, vice versa. Another 3 months went by, and I collected another 15 larvae from each breeding pair. So this experimental design allows me to control for genetic effects, right? I'm comparing larvae from the same parents, just in these two different habitat qualities. So I can really assess the response to these changes in habitat quality. To measure larval swimming speed, we put an individual larvae inside of this swimming flume, which is essentially a water treadmill. We can calculate the U-crit, or critical swimming speed, by the speed of the water that's moving through this flume that we incrementally increase. And then the fish maintains its position in that center chamber until it gets pushed out after it can't handle it any longer, and we stop the timer. So after its little workout, we also then photograph each larvae so that we could measure its length. Also, in case we saw plastic responses in different morphometrics, I also measured body depth, total muscle area, propulsive fin area, but all these traits ended up being really correlated with one another and yielded similar responses. So just for brevity, I'm just going to share the results on total length. Right, so here's the results. We have swimming speed on the y-axis, and each dot is just the average of the 15 larvae collected from each parental pair while it was given the high food ration and the low food ration, right? And each line is connecting for each parent just on the two different treatments. All right, so with these lines, we have a reaction norm framework. And as you can see, there's no obvious trend, right? No matter how you squint your eyes. There's a lot of variation in response to these parents. And then our results from our mixed model also say, yeah, there's no effective treatment on the larval swimming speed. And here's the result for total length. So now total length on the y-axis. And the reaction norm here looks more like what we can interpret as a plastic response, right? Where most of these connecting lines have a very similar response, like across the parents. And from our model, we do find a significant effect of treatment on larval body length, where the offspring that are born to parents during the low treatment are longer than offspring born to those same parents on the high treatment. So that's what I expected in terms of thinking about a dispersal-related trait. But I just want to reiterate, because this is kind of counterintuitive, right? We found larger larvae coming from the poor habitat. So even when you give these fish like less food, they're making bigger babies. So when we think about these criteria, I'm really motivated and excited that, yeah, I think parents can alter their offspring's dispersal phenotype in terms of their baby's larval body size, right? But the next step, of course, is to look for this in the wild and actually measure the dispersal distances of larvae, right? Are larvae that come from these really good environments with like really big anemones, really big moms, lots of food, that they're laying lots of eggs, are these our shorter dispersers? Versus if we see our long dispersers, are they coming from those poor quality habitats? And lastly, to kind of tie it in with the symposium's other topic, right, in response or coping with changing environments, this summer, I'm co-mentoring some undergraduate researchers that are looking at the effect of heat waves and subsequent bleaching on the anemone fish mutualism. And so as we encounter bleaching in the field, we would be interested to see if there is a response in the dispersal phenotype of anemone fish coming from parents that were in bleached anemones. And that was my story. If you have any more questions, you can find me on Twitter. I'm at BU. I really want to give a shout out to my undergrad, Kurt Castro, that very first picture. A lot of swimming trials, my advisor, Pete Buston, and our lab manager, Isa, because aquariums are hard to take care of, and our funding. And if there's time, I'll take any questions. Oh yeah, originally the parents are wild caught, and they've been in the lab for about like 10 years.
Okay, so those are fish that theoretically should have the same starting body condition. Meaning like that first chart that you showed about the first one that had no pattern to it, that one should not have induced a body condition if they were in the lab that long?
No, I'm also comparing offspring like from the same parents. And the original model also includes like a random effect of like the tank ID of the parents and things like that. So yeah.
Thanks for the talk. Is larval length related to clutch size, or do parents have less eggs if you feed them less?
Yeah, exactly. So yeah, when we feed them less, they lay less eggs, right? So I think another way of thinking of this could be like something to do with survival and other things, and maybe dispersal could be like, you know, a secondhand effect of these things. But it was important that we looked at reproduction because we wanted to make sure that the treatment was having an effect on them. So we did record changes in reproduction. And yeah, they lay less eggs and make bigger babies.
Really nice work. If the larvae are larger when they come out, wouldn't that suggest they're closer to open size so they could disperse shorter?
Yeah, there's actually, just to make it more confusing, they're actually finding that better swimmers, larger larvae, maybe that's actually aiding in them having the ability to stay closer. So I think it all depends on like what would be adaptive. So we definitely need to look in the field. Thank you.
Hi everyone, my name is Andrea and I'm a postdoc at the University of Oxford, and today I'm going to present a project that I recently started. So all the results are preliminary and I would appreciate any feedback you can give me. So I will be talking about dispersal and the potential for local adaptation at small spatial scales in a wild-grade tit population. So here I'm just starting with some shapes that can represent either patches in a fragmented habitat or islands in an archipelago. And if we apply different selective pressures, which here are represented by different colors, blue, red, and yellow, what we will see is these populations adapting locally, diverging, and trying to reach their adaptive peak. But natural selection doesn't operate alone in nature. There are other evolutionary forces that can play a role and disrupt local adaptations, such as, for example, gene flow. So in this case, gene flow is represented by these white arrows that are connecting all of these populations. Our alleles are moving from one to another. And as you are seeing here, most of these populations that are connected are nearby, because gene flow tends to happen across populations that are close in space. And that's why we think that local adaptation doesn't happen at small spatial scales that often. It happens at coarser spatial scales. But we have a few examples of microgeographical local adaptation, such as, for example, in California scrub jays, in which we find locally adaptive populations in an island that is quite small. And then also in chaffinches in La Palma, in Canary Islands, in which some of the populations are adapted to more wet and humid forests, and others are more adapted to dry pine forests. And all of these examples are amazing, but they all come, or like most of them, come from islands instead of continental populations. So islands are great because they have like these water barriers that impede the migration from external immigrants, and also there is selection against dispersal ability after island colonization, very often as part of the island syndrome. So we will see more likely local adaptation at small spatial scales on island versus continent. But I wanted to be brave and test that in the continent, so I am going to test for the possibility of local adaptation. So I'm going to be focusing more on the dispersal side of things today. But this is a population of wild great tits that is part of a bigger metapopulation in the UK or the whole of Europe. And this population is fantastic to test these things about dispersal because we've been studying it since 1947. So we have more than 75 years of data. Also, we have more than 1,000 nest boxes in which great tits nest every spring, and we record important life history traits and breeding traits such as, for example, clot size, laying date, and we also ring them or band them. So we put like these individual tags in their legs, and we can know. We can build a social pedigree, and also we can know where they were born and where they have migrated or where they have dispersed for breeding. Also, we can know which ones are the external immigrants because these are adults that we catch in the population that they haven't been ringed or banded. So that is also useful information for these purposes. And also we have associated genomic resources. So these started as a collaboration between Ben Sheldon and John Slade, and we have more than 900 individuals genotyped as with a SNP panel and more than 500,000 SNPs. And these birds were collected between 2000 and 2013. So this is not only the collaboration between Ben and John, but also lots of people that collected this year after year. So I'm going to use these resources to answer some of the questions about dispersal and potential local adaptation. So first of all, we want to know whether there is a spatial structure, because any spatial autocorrelation pattern can explain whether there's a local adaptation or there is limited dispersal. So here I am plotting the first axis of variation of a genomic PCA onto a map of the population in which each point is an individual. And what we see is that there seems to we seem to see some special genetic structure. But now we're testing more for an isolation by distance pattern by plotting identity by descent, which is shared genetic ancestry between two individuals against the dispersal. And what we see is that those individuals that are closer in space also share more genetic ancestry, and there's a decay with distance. So now we want to dig more a bit into like what are the mechanisms underlying this isolation by distance pattern. And when we color the raw data with points by the kinship degrees that these pairs share, we have that those birds that share more genetic ancestry are also parent offspring or full siblings. Similarly, with second degree kinship, and third degree. So if we remove those close relatives, we don't see any isolation by distance pattern, which can reflect limited dispersal. And that fits very well what we know about this system, in which great tits basically disperse more or less 500 meters between where they were born to where they were breeding, and then after breeding, they don't disperse that much either. So now we want to see whether the genetic basis underlying this isolation by distance pattern are erased at some point. So to do this, what I'm doing is I'm getting two sections within the population that are the most distant. So I'm taking like a subset in the north and a subset in the south. And I'm going to compare the identity by descent of pairs in a given year. That is going to give us a nice baseline, which is basically what do we expect in a given year of like the most distance of differentiation. And then I am going to compare as well pairs of individuals within the same year, but that they vary in the year that they were born. So for example, a bird that was born in 2000 with a bird that was born in 2000, 2001, etc. So increasingly separated by time. So we expect that those birds that are more separated by time will share less genetic ancestry. So we will expect to see like a decay that at some point might reach the baseline level. And what we see is that, yeah, we reach the baseline level more or less after 5 or 6 years. So this reveals that the spatial genetic structure is lost at some point, and it is a very dynamic system due to probably population turnover and mortality, but also within population dispersal and external immigration. We haven't talked a lot about external immigration, so we're going to do that now. Here I'm plotting the two first axes of variation of the genomic PCA, and I am coloring the points by immigration status. And what we see is that immigrants are not a discrete group, nothing separated. This is normal because this is a continental population in which there are no real barriers. So we just have some fields outside of the piece of woodland. But still, immigrants can be a bit different. So in this case, I am plotting again the isolation by distance pattern, but splitting it between the three categories. So immigrant, immigrant resident, or both residents. And what I see is that immigrants are less related to each other than residents, which is what we expect, nothing new. Because immigrants can come from the east, from the west, they will share less genetic ancestry than those that are from the population. But that disappears a bit at the end. So here, about like 3,000, 4,000 meters, in which the three groups basically have the same genetic ancestry. So this probably suggests that those immigrants don't come from very far from, they don't come from Sheffield or from Cambridge, most of them. They just come from outside, like close to the population. Then, like, immigrants sometimes can bring some interesting characteristics into the population, including genetic diversity. So what I'm going to do is I'm going to compare the different profiles of genetic diversity and inbreeding in both groups to see whether they differ. So for that, I'm going to calculate rounds of homozygosity. These are identified descent segments, so basically segments in the genome that are highly homozygous. And the length of them and the number of them that we have will reveal how inbred they are. So here what we see is that most individuals don't have rounds of homozygosity, or they are very short. So that indicates that there is not much inbreeding. And then I'm going to do the logarithm of the rounds of homozygosity to get rid of the zeros as well and being able to visualize that nice tail. And what we see is that immigrants and residents don't differ that much. So we don't see much inbreeding, and this is likely due to their high population sizes, both for the immigrants and for the residents. Finally, you know, like when we saw that plot in which I plotted like the two first principal components of the genomic PCA, and there wasn't any discrete group, we can still see at the level of the genome, when we do a genome scan, some parts that could be putatively under selection. So that's something that we can explore because we can have like some parts of the genome that are less or more resistant to gene flow. So I'm going to do that here by comparing two groups. One of them is the immigrant group, and another one is the resident group, in which I am creating a pure line in a way. So I'm getting individuals for which parents, great-grandparents, etc., all of those are resident as well. So then I'm computing FST, and what I see is that there, even though like the genome is quite homogeneous, there are some parts of the genome that are highly divergent, which could mean that there's positive selection, but it could also mean like so many other things, like background selection or many artifacts. So now what we need to know is what is underlying those parts of the genome that are highly divergent, which could be just by doing some selective sweep scans or by looking at the genetic diversity, etc. So that's something that we need to kind of do in the future. So just wrapping this up, first, what we see is that there is spatial genetic structure in the population, but that it is mediated by limited dispersal, by those close relatives that stay in the same spot, more or less. And then that the genetic bases underlying this pattern are renewed from time to time, well, from time to time, not constantly, basically, every 5 or 6 years. And that immigrants are not particularly different to resident birds, both in a genomic PCA, in a global genomic PCA, but also in heterozygosity levels. But still, we see some parts of the genome that are highly divergent, and that could suggest some local adaptation, but still, we still need to look what's behind those peaks, etc. And with that, I want to thank my collaborators, Nilo and John, and my supervisor, Ben, and I'm happy to take any questions. Thank you.
Great talk. Very interesting system you have. I was wondering, I was very interested about your last results, the genome scan between dispersers and residents. And do you plan on maybe looking at like those specific loci, the variation in allele frequency throughout? You have, I don't know, 17 years of data. That would be
Yeah, no, that could be amazing. Yeah, so that's something that we can definitely do. Yeah, so I mean, I was literally like doing those plots like a couple of weeks ago, but yeah, I mean, that's something that I will definitely explore in the future.
I have one question. When you show the map with like the PCA with the colors, it looks almost like a margin versus the middle of the woods kind of patterns.
Yeah.
Do you have time to explore it?
Yeah. So I mean, we know from the population that we can have edge effects in which like the edges are a bit different to the middle, but also that correlates as well with altitude because the center of the population is a bit higher, etc. So yeah, that's something that we can explore as well, and how many immigrants we receive from like and stay in the edges versus like that they kind of migrate into the center of the population. Cool. Thank you.
Hello, good afternoon everyone. I'm Subhasish Halder. Today I'll be talking about habitat fragmentation, sex-biased dispersal, and sexual conflict. What can experiments on mundane fruit flies can tell us? So as you know, human interference not only leads to general decline of the total biodiversity, but it can also fragment population, resulting into population structuring. When I say population structuring, I mean individuals are not uniformly distributed in a population, rather they are structured. They are distributed in multiple subpopulations or demes, which are often connected by dispersal. Although you would expect most of the populations to be spatially structured, habitat fragmentation not only facilitates this structuring, but can also amplify the detrimental effect. This kind of structuring can introduce many noble challenges, noble selection pressure. For example, there can be physical challenges depending on the patch size, patch distance. There can also be demographic or genetic challenges depending on the resource distribution across patches. By now you have realized dispersal is one of the key components that plays key role in this context because it allows individual to move across patches, find the suitable conditions, and thereby increase Darwinian fitness, which is propensity of survival and reproductions. How does population respond and adapt to such challenges has been one of the major interests among evolutionary ecologists. So far, theoretical prediction has been tested by comparing the mean phenotypes between two landscapes which are known to differ by certain degree of structuring. For example, these skippers, there has been shown fragmented population, there is increased investment in thorax and body mass, suggesting resource allocation towards somatic maintenance. This fritillary butterfly from fragmented habitat, there is evolution of dispersal related traits, but that comes as a cost of longevity, suggesting dispersal can interact with other life history traits. In a controlled laboratory setup where dispersal has been selected for, however, there was no associated change with life history. So how dispersal evolve and how it interacts with other life history traits, for example, reproduction longevity, the picture is unclear. We appreciate the fact that dispersal is a prerequisite under the spatial structuring. However, there is an interesting catch. In polygamous species, due to reproductive investment, that is gamete production, and parental investment, the resources that maximizes fitness is differ across male and female. For female, it is important to have feeding, nesting and breeding ground to maximize fitness. However, for males, it depends on the potential number of mates it can mate with. Therefore, depending on the resource, two sexes can always disperse differently, causing sex-biased dispersal. Given this background, understanding the ecological factor that drive dispersal, specifically sex-biased dispersal, and what is the ecoevolutionary consequences with spatial structuring, that needs to be studied with this Anthropocene age. However, by the last two talks, we have realized studying dispersal in the natural population is quite challenging. It has its own confounds and constraints. For example, we do not know the population history. There are logistic issues. Also, getting permission is difficult. And there is statistical limitation as well in terms of replication sample size. I took a bypass. I used a Drosophila melanogaster laboratory adapted population, commonly known as fruit flies, to test some of this problem. So the idea is to create a large founding population collecting flies from the wild and maintain them in the laboratory settings in controlled environment. For example, controlled temperature, controlled light, controlled food, standard density and population size. Since these populations are maintained in lab for more than 200 generations, hence laboratory adapted, you can tweak any of these environmental factors and see how the population is responding. Another advantage is that you can create replicates of such populations and check the robustness of your observation. By using this system, first I tried to understand the dispersal and whether there is sex difference or not, and what are the potential drivers of such dispersal. Following, I wanted to know if certain kind of habitat, that is structured habitat, allows dispersal, what is the fitness consequences. Before going into the details of the talk, I would like to introduce you the experimental setup that I've used. Here's a two-patch setup, common vial which is connected with a corridor, basically a Tigan tube with a sink. If you introduce flies in the source patch, they immediately starts moving towards the sink. Therefore, you can quantify the proportion of flies moving, time taken to move, also if there is sex-specific difference. That means dispersal is actually quantifiable. To understand the consequences of dispersal, I constructed my own structured habitat by connecting 3 vials where flies can move from one patch to another patch. All the experiments I have done with ample amount of replication, which is one advantage of the Drosophila. And depending on the data distribution, either I have used linear modeling or generalized linear modeling. So the first thing I asked, does two sex differ in dispersal? To test it, I introduced 30 flies in the source patch and I allowed them to move for 6 hours. Following 6 hours, I counted the proportion of flies found in the sink. In the x-axis, you see I have female and male denoted in different colors, and in the y-axis, I have proportion of flies moved. Whenever two treatments share the different letters, that means statistically they are different. As you can see, female disperses significantly more than males, and that is quite consistent across the 3 populations that I have tested. Now the question is, why do female disperse? The classic literature suggests that monogamy should favor female dispersal, for example in birds. Polygamy should favor male-wise dispersal, for example in mammals. However, Drosophila is polygamous, but still we see female-wise dispersal. That means there must be some ecological determinants that drive this dispersal. One of the common ecological factor, which is demography, often faced by organism in natural population. If you think about this room as a patch, if female starts moving out from this room, not only the density will change, but also the sex ratio of this room will change. This density and sex ratio can affect dispersal in turn. This is what exactly I tested, how demography affects sex-biased dispersal. In the first experiment, I varied densities in the source patch. I had 3 levels of density control, that means the population regime control, and then lower density and high density. In the following experiment, I varied sex ratio in the source patch. I had 5 labels. I had equal sex ratio, male biased source patch, female biased source patch, all male group and all female group. And I quantified the proportion of flies moving to the sink. This was also done with the sample size. When I analyzed the data, does density affect dispersal? Yes, it does. I found a significant effect of treatment. There's a consistent sex difference. As you see, females disperse significantly more than males. Interestingly, there is a treatment by sex interaction that suggests how density effect male dispersal is different from female. As you see, changing density did not affect female dispersal. However, as the density increased, male dispersal also increased, suggesting that this density can have a sex-specific difference in dispersal. Does sex ratio affect dispersal? Yes, it does, both in female dispersal and male dispersal. As you see, female disperse significantly from a male biased patch comparative to the patch where there are no males. On the other hand, if you look at male dispersal just shoots up when there are no female available. Compiling this suggests that female dispersal is sensitive to the presence of male and male dispersal is triggered by the absence of female. So far the observation is that female disperses and there is enough plasticity. And females also tend to disperse more from a high density male patch. Does it has anything to do with sexual conflict? As I've introduced to the fact that males and females differ in their interest, males often adopt many behavioral strategies which is harmful for female but it's beneficial for the males. For example, this traumatic insemination by bedbugs or the snail. Or there can be infanticide behavior which is seen in many organisms, for example lions, where the male kills off the offspring of the female so that the female is available for mating. So this Drosophila melanogaster has been a standard model that has been used extensively to study the sexual conflict. However, if you have realized, this traditional lab regime differs from natural population by one fundamental aspect, that is dispersal. There is no scope of dispersal in natural population, traditional lab population, but in the special structured population, organism can disperse. There are some evidences that suggest when the environment is relatively complex, that means the male-female free interaction is somehow inhibited, sexual conflict comes down. By extending this finding, I can also hypothesize if a certain population structure, females can move across patches, sexual conflict can also come down. This is exactly what I tested. I created a 3-patch setup which are interconnected. Therefore, flies can move from one patch to another patch, and as a control, they cannot move. I conducted a full factorial experiment having habitat type as one factor, structured and unstructured, male exposure treatment as another factor with 2 levels, limited exposure and continuous exposure. That means in limited exposure, females were exposed to males for shorter duration. In continuous exposure, females were exposed to males for longer duration. The prediction is that if there is sexual conflict, there is male-induced harm, when females are exposed for longer duration, their fitness will come down compared to limited exposure. And this fitness reduction, this decline, the slope of this decline, if that varies across the structured or unstructured, then I can comment that sexual conflict also depends on the habitat type. So I raised flies in standard condition and they were subjected to these conditions. After that, I measured 2 components of female fitness, that is progeny production and starvation resistance. What I found when I looked at progeny production, I found a significant effect of habitat. In structured habitat, females lay significantly less number of eggs compared to unstructured habitat. That is likely because of the movement cost. This movement across patch is energetically demanding. When I looked at male exposure, also has a significant effect. When females are exposed for continuously, which is this brown dot, they lay significantly less number of progeny compared to the limited exposure treatment. That suggests that there is sexual conflict. However, this effect of male exposure did not vary with habitat as I predicted. If I look at the other component of fitness, which is starvation resistance, that was almost comparable. Also, I found a significant effect of habitat, a significant effect of male exposure in the same direction. Again, the interaction was not significant. Compiling this all, interestingly, when I looked at male starvation resistant across these 2 kinds of habitat, that was not different, unlike females. Compiling this all together, there is significant female bias dispersal, there is also plasticity in dispersal. However, this does not affect sexual conflict. Indeed, habitat structure, if certain kind of structuring where there is movement cost, that can have a sex-specific movement cost, and this may act as a term of natural selection. So this is the first experimental evidence in my knowledge, where I have tried to connect dispersal, sexual conflict, and habitat structuring. Future investigation should test this generality of these results by adding more parameters into these equations, for example, patch quality, predation risk, or competitions. What would be really interesting to see that if that structuring and that movement cost is a persistent aspect of natural ecology, what is the long-term consequences? How do the female evolve? If you are interested, so this is the end of my talk. If you are interested to read more about it, this is already, some of these data are published in Linnean Society. Last but not least, I'd like to thank my guide, Dr. Bodhisattva, and my lab members whom you can visit by scanning this, my host institute, my grant, and specifically sir to allow me coming here by giving me money. Thank you, and I'm open for questions.
Did you look at the relatedness between the flies and whether more related virus of females are more likely to trigger stronger dispersal?
So can you repeat that?
Yes. Did you look at the relatedness between females? And if, in principle, if you have more related females, they should try to disperse more. That means they are more unrelated.
Well, so the thing is that these populations I was talking about, they are maintaining discrete generation cycle. Therefore, one generation do not meet the other generation. Therefore, there is no requirement, there is no kin interactions or.
Yeah, but relatedness can happen between sisters. It doesn't have to be mother-daughter or sister-brother.
Oh, that I have not looked at, how relatedness affect this pattern. Oh, sorry, yeah.
I've seen a couple of papers talk about complicated environments and reduction of methane in those situations.
Yeah.
Do you think this lies, I might have missed it when you were describing, but do you think this results contradictory to those findings?
Oh yes, because this study, when I say they are complex environment, there is basically females can hide or there is options of refuge. That does not need active movement. However, if you allow active movement, that is movement from one place to another place, dispersal, which is energetically demanding, that might change the picture, so it might not reduce the sexual conflict. Thank you.
Hi, good afternoon, everyone. I'm Kelton Cheung, a PhD student in University of New South Wales, Australia. So today I'm going to talk about something about cane toad. So you may have already heard some talks about how species or natural population adapt to changing environments. But I would like to let you know that not only them, but also invasive species also face a lot of environmental challenges. They often intentionally or on purpose or accidentally brought to different places in the world, but they cause a lot of trouble on the ecosystem or the economy. But on the other side, they are a good model system for studying rapid evolution because they often adapt in response to new environments in a very rapid manner. So, and also it provides insights on the evolutionary mechanisms that allow them to behave so successfully. And in my PhD, I primarily focus on the guy, this one, the cane toad, Renella marina, one of the most well-known invasive species in the world. The reason why I picked this species is because of their ecological significance in Australia. They produce lethal toxin that kill many reptiles and animals in Australia already. That's why we have a rich body of research about the evolution of the ecological and physiological changes after they're introduced into Australia. So let me give you some introduction history of this cane toad. So they are native in South America, but in particular this invasion, they are taken from Guyana and French Guiana into a number of Caribbean islands. And in early 19th century, they were taken to Puerto Rico. And in 1932, some of the individuals were taken to Hawaii as a pest control. 3 years later, 101 individuals were taken to Australia also as a sugarcane pest control, but they failed to do that, as you all know. And like many other invasions, a lot of studies focus on the invasive range to learn about how these species adapt. So after the toad were introduced into the wet tropic of the Queensland, and they expanded their range westward to the dry desert in the west side of the country. When we compare the toad on the wet tropic to the dry desert, research found that their morphology, physiology, and behavior differ a lot. And some of these changes have been shown to be plastics responses. While other changes may be due to novel mutation, it's more likely that they result from standing genetic variation from native range. That's why it's important to study the toad genetics in the native range. So our collaborators have studied the toads in French Guiana from the sites shown in red. If you see the yellow desert logo next to the site, which means that the site is dry in environment. If you see the blue rainy cloud logo next to the site, which means the area is wet. So they found that the toads are much bigger in the rainforest than the coast. And in the rainforest, you can see that the toads they can be easily access fresh water in the puddles. But while on the coast, you can see that the toads are just burying themselves in sand and then prevent dehydration from the sun. And they have limited access to fresh water. So you can think of the beach as a linear desert. So it's also interesting that we see the sex ratio in rainforest is male bias. So we want to know if there's any genetic basis of the morphological difference. So that's why we use the whole genome resequencing data of 3 individuals from the rainforest and 3 individuals from the coast. And we can see that there's large differences between these groups. In this PCA plot, we have also included some invasive cane toads here, and it seems that they are genetically intermediate. In another study that used a separate SNP panels of 34,000 SNPs, also found differences between rainforest and coastal toads in both Guyana and French Guiana. So this collection of evidence makes us wonder if Australian toads may have enriched standing genetic variation if they contain allele from both environments. So in this project, there are 2 aims. The first aim is we want to sequence more native toads to see if we still see the same pattern that we just found in the handful of resequencing data. So we sequenced 48 more native toads across the country using dark-seed, which is a reduced representation sequencing technique, and conduct some population genetic analysis. So the second aim is if we hypothesize that if the toad were taken from both coastal and rainforest environment to the Caribbean, it's very likely that they were a mix there. In that case, the toad ended up in Australia may carry alleles from both coastal and rainforest environment. So the allele that increase fitness in the dry area in the native range are likely to be favored in the arid area of Australia. We might see that the patterns, like the alleles, will be less common in the wet area in Australia, but increasing frequency if we look at the toads from the dry area. So in order to test this hypothesis, I will examine how the allele frequency changes across the invasion trajectory. But some of you may know that it's a little bit tricky to pick a allele frequency threshold to see whether the alleles are adapted to the environment. So to make this case simple here, so I'll only pick the alleles that are completely fixed in either rainforest or coastal toads. So here is a frequency graph that we expected to see, and it shows an ideal situation where for an allele is completely fixed in the coastal toad in native range, but absent in the rainforest toad in the native range. And you can see an increasing frequency when you look at the toads in the invasive range when they're taken from wet area to the dry area. So let's look at some results. So after the sequencing and filtering of the SNPs, I ended up with 50,000 SNPs, and then the annals onward, I'll keep using that data set. So what we expect in a PCA, that because I wanted to use the PCA to see how the individuals are genetically differentiated, I expect that I will see the rainforest toad and coastal toad, they form different clusters on the PCA plot. So when we look at all the individuals that I have sequenced, the invasive toads and the native toads, they are separated on the PC1 axis that explain almost 30% of the variance. And if we only look at the native toads, it's pretty good that we still see that the toads are still separated by the environments that they live. But we can see some substructure of the toads that live within the environment. So you can see that the toads are separated according to the geography location that we sampled there. So they form different clusters on different PCA axes. So it's supported by a significant isolation by distance matrix. And this result suggests that if we look at the genotype difference of the toad from different environments, it could be due to the selection based on the environment, or it could be just simply the strong genetic structure that we see in the PCA plot. So we need to be mindful with that when in the downstream analysis. So let's look at the result of the second aim. So I'll be showing some frequency graphs, and this is a short recap of what we expected to see. So for the alleles that are fixed, completely fixed in the coastal toads in the native range, we expected to see an increasing trend if we look at the toads from the invasive range. And for the alleles that are completely fixed in the rainforest toad in the native range, we expected to see a decreasing trend in the invasive range from the wet area to dry area. Okay, let's jump to the result. It's a little bit messy, but it's fine. I will try to explain it clearly. So here are the allelic frequency changes of the SNPs that are fixed in the rainforest toad in native range. And I have removed the SNPs that are invariable in the invasive range because they cannot be under selection. And the colored line here means that the alleles that are fixed in the rainforest toad in the native range, and they fit our prediction in the invasive range. And of these alleles, there are only less than 1% of them out of total fit our prediction. And only one locus that maps to an annotated gene region of the genome. In contrast, if we look at the alleles that are fixed in the coastal toads in the native range, we found a much more alleles that follows our prediction in the invasive range. This may suggest that the coastal environment in the native range have a stronger selection on the toads. And of these alleles, I found 28 annotated genes. They are located in 28 annotated gene bodies. Unfortunately, when I look at the gene ontology term, there is no overrepresentation of any terms. They're just general metabolic processes or cellular components, which cannot comprehensively explain the differences that we see on the toads between the environment. So next, I have done some selection analysis using two different programs. One is Bayescan, which looking at the diversifying selection between the environment. And the second one is the BayPass, which contrasting the environments, different environments in the native range. For the Bayescan, I found 9 candidate loci under selection. And I have looked at the frequency change in the invasive range. Unfortunately, they're either fixed or they didn't show any trend. But it's pretty, it's really expected because the introduction have undergone multiple bottlenecks. These alleles may be just gone in the introduction. So for this, for BayPass, I didn't find any puberty of candidate loci under selection. Overall, this result is a little bit disappointing, but still within our expectation, because we are only dealing with a reduced representation genetic data set, that we are only looking at a tiny bit of the genome that we may just simply miss the signal that we hope to see. So next step will be looking at the whole genome data set to see whether our hypothesis still stands. So overall, we found that the native range toad, they differ in morphology and also genetics, and they have a very strong genetic structure. And the invasive toads, even though I haven't spent a lot of time in this talk, but they still seem to be genetically intermediate. And also the coastal environment in native range seems to be pose strongest selection pressure on the toads. And lastly is we are now waiting for whole genome resequencing data to come back, so it's time for me to work on more. Lastly, I'd like to thank my supervisor, lab members from the Rolling Stone group, and particularly I want to thank the members from Team Buffalo because they did all the field work in French Guiana, and also the funding body that support the work on all the Canto awards. Yeah, thank you so much.
So in considering the effects of standing, I'm sorry if I missed this, but in considering the effects of standing genetic variation versus new mutations, it seems like this would be a situation where there could be really strong founder effects from the group that was brought over. I guess how are you guys thinking about that and looking at that, or do you have an idea of the makeup of like the actual founders from the Australian population?
Yeah, but because in this project we are not, our focus is not trying to find which population are the founding population of the introduction. We're trying to see if because the toad, they are coming from different, they are coming from different environments and whether they contribute to the adaptation here. But definitely there's some alleles that just arise after the introduction because all the scenarios are just happen at the same time in this system. So we will look at all the possibility if I can, yeah.
Thank you for your talk. I'm wondering if you've looked, since there's clearly some spatial structuring too, if you've looked at those potential like outlier loci using any program that accounts for that spatial structure. If you do that, do you still see like no candidate loci under selection?
Yeah, BayPass actually will account for the population structure, but here we didn't see any, so that's why I said, because we may just miss the data from the reduced representation data set. So that's why we will work on the whole genome data set later on.
Thank you for the talk. I have a question. So for the founder population, you took the genotype of modern toad, right?
Sorry, we can talk later. How about that?
Hey everyone, my name is Louisa Bartkovich. I just finished up my second year at University of Toronto in the Bontrager lab. And today, with the help of Clarkia pulchella, we are going to be building an understanding of how plants may be responding to their environment through flowering time. Just so everyone is on the same page, flowering time is an important trait for plant fitness as it allows individuals to synchronize with favorable environmental conditions while possibly avoiding pests and pathogens. Plants have been known to leverage this trait in order to respond to their environment in different ways. Through plasticity, plants have been found to change their flowering time in response to their soil microbiome. Through movement, even though plants cannot get up and just walk around, they actually manipulate patterns of gene flow, which is a type of movement, by determining when and how pollinators can have access to their pollen. And lastly, through adaptation, plants can actually align flowering time with their environmental conditions, leading to flowering time actually aligning with climatic conditions. I'm exploring all of these in my PhD and how they can facilitate persistence in the face of changing climates, but today I'm going to be talking about flowering time and climate in particular. So to start with a very simplified model, you may expect that across a latitudinal gradient, the environment will basically be pretty easy to follow. For example, going from the north to the south, it may become warmer and more dry. However, if you have varied topography, so let's say different levels of elevation, it could make it so climate no longer aligns with the latitude that you're looking at. Additionally, differences in seasonality can mean that populations may experience different patterns or different speeds under which they may ramp up to a season such as summer over here. And so with all of this, and understanding that sometimes climate will not align with a proxy such as latitude, it made me ask which components of these complex climatic landscapes do populations actually adapt to. So I examined the role of flowering time in populations of Clarkia pulchella as an indicator of possible local adaptation. And to do this, I asked 3 questions, all of which build on each other to answer this question. The first one, in a common garden, do populations display differences in their flowering time coming from different climates? And this allows us to know whether or not these differences are actually heritable. Second, does climate of origin explain these variations in flowering time? And again, this is basically an indicator of possible local adaptation in these populations. And lastly, which specific climatic conditions are important in determining flowering time? And understanding the environmental conditions that may lead to populations evolving later or earlier flowering time is again important to understand how populations may then adapt to changing environments in future climates. So this is Clarkia pulchella, and to explain what's going on with Clarkia pulchella so everyone's on the same page, it is a winter annual wildflower that first germinates in November and overwinters as a seedling before undergoing vegetative growth from March to early May, and then undergoing reproductive growth and flowering in late May to early August. And these populations are found in sparsely vegetated hillsides and mountainsides in the Pacific Northwest. And these are just 2 photos of where you can find Clarkia pulchella. And for my experiment, I took field-collected seeds from 17 populations that span from the northern to the southern edges of this range. And they are representative of the topography and the climate of this range. And just hearkening back to my introduction, you may notice that some of these climatic conditions don't really seem to be fully aligning with latitude due to the topography of the range. I'm going to get into that later, so you don't have to fully understand that now. So I took these seeds and I grew 950 plants and collected date of first flower for 866 of them. Let's get into this first question of whether or not these populations are actually varying in their time of first flower. So on this graph, you'll see that for the y-axis, that is days to flower, and for points that are found higher up on the screen, that means they're flowering later, and points lower down to the screen are flowering earlier. And then I just have all of the populations across the x-axis right now just in alphabetical order. So the first thing you'll notice is there is variation in the timing of first flower. And when we then look at latitude, which now we organize the populations with northern populations found closer to me, you'll see that they are flowering later. And these differences are about 68 days between the earliest and the latest population. And that's more than 2 months. So latitude is explaining some of the variation, but it's not explaining all of it. Right now, I've just highlighted these 2 clusters of populations, and you'll see that stuff is not actually following exactly latitude, which may mean that there's something going on with climate. So now we see that there is differences in flowering time with these populations, and it's not being fully explained by latitude. Let's now look at these next 2 questions to see if we can find if flowering time does actually align with climate. So I decided to look at 2 biologically informed variables. One is drought exposure or climate moisture deficit, and then also mean annual temperature. And at first I looked at the annual patterns of these, which you can see in these heat maps. But as I started to explore this even more, I found that there was some temporal variation throughout the year. And when you're looking at temporal variation, so let's look at temperature, you may start to look like this when you meet with your supervisor, when you're trying to talk about time, space, all of that. So to go over this with y'all, so I can avoid looking like this a little bit, I'm going to be walking you through the year. So we're going to start with November, and you'll see in the plot furthest away from me, that's looking at temperature and latitude. And basically points that are higher up are populations that are warmer. Then the plot closest to me is a heat map, basically representing the same thing, but through space throughout the range of Clarkia. And then I also have a Clarkia cam up in the corner closest to me, just to remind everyone about what's going on with the plant during this time. And I have a line of best fit, basically trying to show y'all what's kind of going on with the pattern throughout the landscape. So in November, December, not really a trend. January, not really a trend. February, again. March, it starts to see like the north is heating up a little bit. Same thing in April, there's starting to be a pattern. And really in May, you see this trend that the northern populations are warming up quicker than the southern populations. And then in June, the south starts to catch up and then overtakes the north in July. And this is still happening, that the south is still on average warmer during August. And this is during that time is when Clarkia is starting to flower and has finished flowering by August. So we've now looked at temperature and we saw that there's this interesting pattern across time, across the year. Now let's do the same thing with drought, but instead of showing you all the same types of graphs, we're instead just gonna block it of months before flowering and months after flowering. And I'm just gonna be putting drought exposure on the x-axis and then latitude on the y, with populations found higher up on the screen or more north. So again, March to May and then June to August. And you'll see that in March to May, there's not really a pattern, but then in June to August, which this is just a line, not a line of best fit, you'll see that northern populations are on average less dry than southern populations. So everything's getting dry, but the south is getting a lot drier. So now I have all of these variables that are pretty correlated throughout the year with each other. And so in order to reduce that dimensionality and actually analyze and visualize that relationship, I stuck everything into a PCA. And once I did that, you get a lot of this information, and you guys don't need to understand all of that. I'm gonna walk you through the PC1 and PC2. So for the PC1, stuff further away from me, so more negative, is going to be annually cooler and dry. So that's just the annual trend. And stuff closer to me, so more positive, is annually warmer and very dry. Then for PC2, that's getting into the seasonal differences. So stuff that's positive, or going towards the top of the screen, has an extreme and sudden onset of a drier summer, while points that are negative are going to have this gentle ramp up to a dry summer. So now that we have PC1 and PC2, let's see how they might explain flowering time. So for the first one, let's look at PC1, which is again the annual patterns. And when we do that, we don't really see any trends. It's not explaining the variation in flowering time. Let's do the same thing with PC2, which, just to remind y'all, points that are closer to me, so populations closer to me, have an extreme sudden onset of a drier summer, while populations further away from me have this gentle ramp up to a dry summer. So when we do this, we do find that there is some explanation. So basically, the divergent patterns of the summer transition is explaining the flowering time. And we're seeing that populations that originate from these sites that have a gentle ramp up are flowering later, and populations from a more extreme and sudden onset are flowering earlier. And again, this difference that we're seeing, at least in the greenhouse, is a difference of 68 days, or more than 2 months. So in summary, we found that flowering time is varying across these populations. We also found that climate of origin is important, specifically looking at both annual and summer transitions. But the summer transitions are the ones that are explaining the most amount of this variation that we're seeing in flowering time. And so overall, it seems like populations of Clarkia pulchella are adapting in response to these seasonal changes within their local environment rather than their broad climatic trends. So what does this all mean? Basically, when you're looking at anything to do with changing climates, you may want to explore seasonal and your annual scale. So when you're looking at these populations, you may think for something like assisted gene flow, we may want to take populations from more southern, warmer on average annually, up to these northern that are a little bit cooler. However, you may want to also be looking at the summer trends and seeing what is going on there. So for my next steps, I'm really excited to be running a growth chamber experiment next year that's going to be looking at the plasticity and adaptation of vegetative and reproductive traits in Clarkia pulchella. I'm also going to be leveraging herbarium specimens to broaden the spatial and temporal aspects of these patterns. And that's the end of my talk, and I just want to say thank you to the amazing undergrads in this lab. Everything I do would not be possible without them. I also want to really highlight Tara, who drew a lot of the stuff that you saw in the slides. She's an amazing illustrator and also an amazing scientist. I'm going to get to Ellie later, don't worry. And for my acknowledgements, I am the product of just an amazing community that I am at University of Toronto, and I really want to thank Megan for being an outstanding supervisor for the past 2 years. I look forward to working with you for a lot longer, hopefully. And I also want to thank Davidson College. I was a Smith Scholar last year, and they paid for my housing and food, which was amazing. And lastly, if you want to see an amazing undergrad project, y'all should definitely go to the poster session after this and go talk to Ellie about Trillium and the disease that is impacting it. So yeah, any questions?
Any other phenology measures like heat or duration? And then also, did you do anything like count the number of total flowers or seeds or anything like that?
Yeah, no, that's a great question. So just so everyone can hear that, basically, did I collect anything about overall flowering phenology, so not just the initiation of flowering? Did I collect anything about the duration, the number of flowers that were produced, all of that? So that was not part of the experiment. So I instead just looked at the initiation of flowering, and I collected all of the plants afterwards, so later on if I want to count fruit and everything. But I also did a bunch of morphological measurements at the time of flowering. So I have another project that I didn't talk about here, which is right here, is I found differences in size between when they flowered. And so I have some stuff about early vegetative growth rate and also their overall height and number of leaves and nodes at the time of flowering. And hopefully I'll be able to look at that data and analyze it. Yeah.
Thank you for the talk. It was super nice. I just have a question on your model with the axis
Yeah.
position of the axis. Did you check for the interaction between both? I don't know if you don't have this type of environment
Yeah.
with all combination of slow and fast increase temperature and growth.
So do you mean interaction between PC1 and PC2?
Yeah.
Yeah, so I checked the correlation coefficient just to make sure that they're not correlated whatsoever. And when I actually ran the model, it was doing the independent interaction controlling for each one. So it was PC1 plus PC2. Yeah, in the model.
Do PC1 and PC2, you didn't check for the model, the interaction between the axes?
No, just because I wasn't finding that there was a correlation between the two of them, so I didn't see a point of doing an interaction between the two of them. Yeah. Any other questions?
I guess related to the question that was over there, so
Yeah.
you probably said this and I might have missed it. Did you quantify flowering time as days from germination to first flower or days from planting?
Yeah, no, that's a great question because I did not go over that at all. It was time from germination to flowering, so it was days to flower. Yeah.
Interesting, we find the opposite pattern of latitude and
Yeah, so are you looking at an eastern plant? Okay, cool. Yeah, because it was something I had to wrap my head around is stuff in the south, west, or northwest, anywhere on the west coast, is going to be driven a lot by drought. And so with this specific populations, as you're going more south, you're getting more droughty. And so that may lead to stuff like drought escape, that they might flower earlier. But again, this is in a common greenhouse, or a common garden in a greenhouse. And so because of that, I can't say anything about the timing of germination out in the field. So it could be that these populations are actually germinating at different times, so they actually flower at around the same time. But yeah.